Hello everybody. Now we are going to discuss transport across cell membrane. Okay. So first thing is that you have to remember two concepts. One, that if the molecule is a lipid soluble molecule, okay. So lipid soluble molecule, or if the molecule is a non-polar substances, then it can easily pass through the cell membrane by the process of diffusion. Okay, so the transport is easy by the process of simple diffusion. Like for example, oxygen, carbon dioxide transport, everything, they are the non-polar substances. Simple diffusion occurs for these gases. But the problem arises when the molecule is a charged molecule. So, if the molecule is a charged molecule or if the molecule is a large molecule, then the cell membrane is almost impermeable to them. And in these cases, what you require? You require certain transporter protein, or you require the process of endocytosis or exocytosis, right? So we'll be discussing all these things: transporter, endocytosis, exocytosis, everything. So first, let's discuss about this transporter, right? So you have to remember the transporter molecule, they can be of two types. Transporter, they can be of two types. One is known as the carrier protein, carrier proteins, and second one is the channel protein, channel proteins, right? So carrier and channel protein, they are different, very, very different, right? So you have to understand the concept first. Carrier protein, suppose I am drawing one plasma membrane here, okay. On this plasma membrane, this is the carrier protein with a particular shape. And suppose this carrier protein is going to transport glucose from one leaflet to the other leaflet of the membrane. So what will happen whenever glucose attaches with this carrier protein? Okay, what will happen? The carrier protein is going to change its shape first. So this is the change in shape. Because of the change in shape, what will happen? The glucose molecule will be exposed on the other side of the cell membrane, and then the glucose molecule will be released from here. Getting my point? So what I told, carrier protein means it will attach with the substances which is going to be transported first. Okay, so binding. Okay, so first it binds with the substances which is going to be transported. Then the carrier protein molecule is changing in shape. Okay, change in shape. Then it is changing in shape, and then it is releasing the molecule on other side of the membrane. This typical sequence is going to happen in all of the carrier protein molecule. The carrier protein is required not only for active transport, but it can also require for passive transport, particularly for facilitated diffusion. I'll be discussing this in detail. But for the time being, you remember carrier protein means, okay, they are helpful for not only for your active transport, they are also helpful for passive transport like facilitated diffusion. But if you look at the channel protein, okay, so if this is your plasma membrane, okay, and on this plasma membrane, suppose here there is a channel protein. There is a channel protein present here. This channel protein, this contains one pore inside. Through this pore, the molecule will pass through directly from one leaflet to the other leaflet of the membrane without any attachment with this channel protein. Getting my point? Suppose the sodium is passing through this channel protein. So sodium is not going to interact with the channel protein per se. Sodium will pass through this protein molecule because there is an intracellular pore. There is a pore inside, not intracellular. There is a pore inside this channel protein. Through this pore, the sodium will pass through, right? And obviously, the channel protein is not going to change in shape, although certain channel protein contain certain gates. Okay, so these are known as the gate of the channel protein. Now, how this gate will open up in case of channel protein? It can open up in presence of voltage change of the membrane. Suppose this cell membrane is depolarized, right? So what will happen? The gate may be open up. That kind of channel is known as voltage gated channel. This is known as voltage gated channel. So the gate is opening in presence of voltage. Which voltage? Depolarizing kind of voltage. Or suppose some hormones, some neurotransmitter is attached on this channel and then the gate is opening. So this is ligand. So this is known as ligand gated channel. This is ligand gated channel. Or some of the channel, they open up in presence of stretch on the membrane. When the membrane is stretched, suppose this membrane is stretched on this, okay, what will happen? The channel may also open up. That is known as stretch sensitive channel or mechanosensitive channel. Stretch sensitive or mechanosensitive channel. Okay, so these are the different variety of channel. There are many others also, but these are three different types of channel protein that you have to remember. Okay, so the channel protein means it is not going to change its shape and it is not binding with the substances which is transported through it. But carrier protein, it has to bind with the substances and it has to change its shape. Whenever any transport occurs through carrier protein, it will follow three kinetics. Number one, saturation kinetics. You all know this thing. Suppose there are two carrier protein here and there are four substances present in this. So what will happen? All the carrier protein will be saturated with the substances. Okay, so that is known as saturation kinetics of carrier protein. Okay, second thing is that specificity, specificity. Okay, you know the D glucose and L glucose, you know, if the carrier protein is dedicated for a particular isomer of molecule, then it will not be able to transport the other isomer. Like for example, if the carrier protein is dedicated for D glucose, then it will not be able to transport the L glucose. That is known as stereospecificity of carrier protein. Okay, so saturation, stereospecificity, and number three is competition, right? If the carrier protein is dedicated for transport of two similar kind of molecule, like for example, there are carrier proteins which are dedicated for transport of glucose also and galactose also. So what will happen? Obviously, there will be some competition between this glucose and galactose. If the glucose concentration is high, that will occupy the carrier protein, and if the galactose concentration is high, that will occupy the carrier protein, and transport will occur for galactose only. So this is known as competition. So please remember, whenever we will say that this transport process is based on carrier protein, it will also follow three kinetics. One is saturation, one is competition, and third one is stereospecificity, right? So this is about the carrier protein and channel protein. Now we are going to classify the different types of transport process here. So now we are going to classify the different types of transport. Okay, so all of you know this classification very well. Okay, type of transport: active transport, passive transport, and there is a separate one for large molecule, that is endocytosis and exocytosis. Okay, so first we will be concentrating on active and passive transport. So all of you know, active transport means this is energy dependent transport. You require source of energy in the form of adenosine triphosphate, right? So two things: one is energy is required in the form of adenosine triphosphate, and second thing, active transport means transport will occur from low solute concentration to high solute concentration. This is known as against the concentration gradient, or this is also known as uphill kind of transport. So please remember, the active transport means you have to remember two things. One is it required the adenosine triphosphate, and this is uphill kind of transport. It occurs from low solute to high solute concentration. And the opposite is true for passive transport, means no requirement of energy is here, means adenosine triphosphate is not required, and transport will occur from high solute concentration to low solute concentration, high to low solute concentration, that is known as downhill kind of transport. So no ATP is required, and downhill kind of transport, that is typical of passive transport. Okay, now active transport, again it is divided into two. One is primary active transport and one is secondary active transport. Secondary active transport, primary active and secondary active transport. Secondary active transport is again subdivided into symport or co-transport and antiport, okay, or counter transport. Symport or co-transport, antiport or counter transport. Now first you look at this primary active transport. So remember, primary active transport means, in books it will be written that primary active transport means when the energy utilization is direct. Energy utilization means when this ATP use is direct, then that is known as the primary active transport, and when the adenosine triphosphate use is indirect, that will be known as the secondary active transport. So you have to understand what is the meaning of this direct use of energy and indirect use of energy for primary and secondary active transport respectively. That we have to understand. So first you remember that primary active transport means, if I give example, any transporter, any carrier protein where the name is attached with ATPase, all of you know this thing, sodium potassium ATPase. So whenever any carrier protein, so this is a carrier protein only, okay, getting my point? Carrier protein means it will have the three property: saturation, competition, stereotoxicity. So when any molecule is attached with this name ATPase, then that is a primary active transporter. ATPase means it can break down the adenosine triphosphate molecule. That's why ATPase, right? And the example of the primary active transporter, the universal pump which is present in almost every cell in our body, is sodium potassium ATPase. Now first we will look at the structure of this sodium potassium ATPase, then we will move on to the other part. So this is the structure of sodium potassium ATPase. What you are finding out? You are finding out two subunits, beta and alpha. So basically, you have to remember that sodium potassium ATPase is a heterodimer. It is a heterodimer composed of two subunits, alpha and beta. Now we look at the beta subunit here. So this beta subunit contain three glycosylation sites. So this is 1, this is 2, this is 3. What are those? This is glycosylation site. With the help of this glycosylation site on beta subunit, the sodium potassium ATPase molecule remain attached with extracellular matrix protein. So this is suppose extracellular matrix. The sodium potassium ATPase remain attached with extracellular matrix protein with the help of beta subunit, with the help of glycosylation, which is present on beta subunit, right? So I remember like this: beta for binding subunit, it is remain attached with extracellular matrix, right? All other functional aspect of sodium potassium ATPase pump is due to alpha subunit. Means basically you can understand like this: the alpha subunit is the regulatory subunit of sodium potassium ATPase pump. Now you look at this molecule more closely. On the intracellular surface, means on the ICF site, there is a site for attachment of sodium. That is the point number 1, sodium attachment site. So what will happen? Whenever the molecule is going to transport 3 molecules of sodium and 2 molecules of potassium, all of you know that thing. First, the sodium as well as the adenosine triphosphate will attach to the alpha subunit simultaneously. What I told? This sodium as well as 1 molecule of adenosine triphosphate, both will binds. ATP binding site, okay, is there. So both will bind simultaneously on the alpha subunit inside the cell surface, inside the intracellular fluid. Now after binding, what will happen? Because the molecule contain ATPase activity, this adenosine triphosphate molecule will be broken down to adenosine diphosphate. And the phosphate will be transported. Phosphate will transport here. The phosphate will transport in the phosphorylation site. That means I can understand that whenever you break down the adenosine triphosphate to diphosphate, there is phosphorylation of one of the amino acid in this molecule. Because of this phosphorylation, okay, so whenever there is phosphorylation, there is change in shape of this sodium potassium molecule. There is change in shape of this sodium potassium ATPase molecule, because of which what will happen? This sodium will be thrown out of this cell. Getting my point? I told you already that carrier protein changes its shape, and because of the change in shape, the sodium which was attached inside, now the protein has changed in shape, and that's why the sodium is moving to outside. So phosphorylation is changing the shape, and that is throwing the sodium out of the cell. So far, potassium has not attached with the molecule. Now the potassium is going to attach. So now this potassium molecule is going to be attached on the outer surface of the protein molecule. Then what will happen? There will be dephosphorylation, and because of the dephosphorylation, the molecule will again return back to the original shape. Again return back to original shape, and whenever it is returning to original shape, what will happen? The potassium will move inside the cell. right? So whenever potassium binds with the sodium potassium ATPase molecule, dephosphorylation will occur and potassium will move inside the cell. When sodium binds with the molecule, along with that ATP also binds, adenosine triphosphate also binds, and phosphorylation causes change in shape and sodium thrown out of the cell. So this way the sodium potassium ATPase pump work, right? Now you also have to remember that this pump is blocked by drugs, which is Wabens and related digitalis groups of drugs, which is a blocker of the sodium potassium ATPase pump. And look at, the Wabin has an attachment site which is on extracellular surface of the ATPase pump. Okay, so Wabens and digitalis groups of cardiac glycosides, they are the blocker of this ATPase pump, right? So this much information you have to remember for sodium potassium ATPase pump, right? So during this discussion, you have already noted that the carrier protein, that is a sodium potassium ATPase pump, it's a carrier protein. Yes, it's a pump, it's a carrier protein. This carrier protein has ATPase activity, and because of the ATPase activity, the molecule is capable of breaking down the adenosine triphosphate molecule itself. That's why we say that primary active transporter means there is direct use of energy. There is direct use of energy. What do you mean by that? Means the transporter molecule, the carrier molecule itself is breaking down the adenosine triphosphate molecule. Direct use of energy. Now you look at the secondary active transport. I hope this is clear, primary active transport. Now we look at the secondary active transport. I told you secondary active transport are of mainly two types, symporter and antiporter. Example of symporter is SGLT, sodium glucose linked transporter, and antiporter, the typical example is NCX, sodium calcium exchanger. Exchanger is written like, exchanger is written here with X, sodium calcium exchanger. So symporter typical example is SGLT and NCX. So we'll draw a diagram and we'll try to understand this thing. Okay. So I'm drawing one intestinal epithelium cell here. Intestinal epithelium cell here. Okay. So this is the lumen of the intestine. On this luminal aspect, the transporter which is present here is SGLT. Okay. Whether it is one or two, I will tell you later. But suppose this is a carrier protein which is transporting glucose as well as sodium. That's why this is known as SGLT, sodium glucose linked transporter. Now we look at, first of all, glucose and sodium, they are moving together. That's why they are known as a symporter. Now sodium is moving from high concentration to low concentration, means sodium is moving along the concentration gradient, but glucose is moving against the concentration gradient, because in the intestinal cell the glucose amount is very, very high. Okay, so from lumen to intestinal cell, when the glucose is entering, it is against the concentration gradient, but sodium is moving along the concentration gradient. So what is the definition of active transport? The active transport, energy is required, and second thing is that transport will occur from low solute to high solute concentration, means against the concentration gradient. In this symporter, we are finding out between these two molecules, sodium and glucose, only one is moving against the concentration gradient, and that is correct, that is true. Okay, not all the molecule has to be transported against the concentration gradient. At least one of the substances has to be moved against the concentration gradient. For sodium potassium ATPase pump, we have seen both sodium and potassium moves against the concentration gradient. But in case of SGLT, glucose is moving against sodium is moving along the concentration gradient. And because of the movement of the sodium, glucose is also moving. Alright. So, suppose you are absorbing more and more glucose from intestine. So, you are absorbing more and more glucose from intestine. So, what will happen? Parallelly, your sodium concentration within the cell will also rise. Now, your job is to throw this sodium out of this cell. Why? Otherwise, if the sodium is accumulating inside the cell more and more, then sodium will not be able to move from outside to inside. And if the sodium is not moving, glucose will also not be able to move. Right? So, I have to keep the sodium concentration inside the cell low so that this SGLT able to work. Right? So, how can I throw sodium out of the cell? You require the universal transporter protein, that is sodium potassium ATPase pump. So now I can understand that this SGLT molecule, it is dependent on the sodium potassium ATPase pump. Getting my point? So this SGLT molecule is dependent on this sodium potassium ATPase pump. And whenever sodium potassium ATPase pump work, we know that it breaks down one molecule of adenosine triphosphate to adenosine diphosphate. That means I can understand even this SGLT require energy, but this energy, okay, where from it is getting energy? It is not breaking down the adenosine triphosphate molecule itself, but for the energy source, this SGLT molecule is dependent on the sodium potassium ATPase pump. Getting my point? So always remember, the secondary active transporter is always dependent on primary active transporter. Secondary active transporter is always dependent on primary active transporter. If you block this primary active transporter, sodium potassium ATPase pump, your glucose absorption will be prevented from lumen to cell. Getting my point? Because SGLT is dependent on sodium potassium ATPase pump. Right? That's why the energy is utilized by sodium potassium ATPase pump, but SGLT is dependent on that. That's why secondary active transport means use of energy indirect. So indirect use of energy. Indirect use of energy. Right? This is the meaning of secondary active transporter. Correct? Okay. Now, again we will take another example. That example is sodium calcium exchanger. So, I am drawing another cell here. This is typical of myocardial cell. This is typical of myocardial cell where calcium enter inside the cell during action potential, and this calcium causes contraction. This calcium causes contraction. After the contraction of the myocardium is over, my job is to throw this calcium out of this cell so that the myocardium will be relaxed. Right. So I have to throw this calcium from inside to outside the cell. What do you think? This movement of calcium from inside to outside is downhill or uphill? It is uphill kind of transport, because you know the extracellular concentration of calcium is enormous and intracellular calcium is in nanomole. So if you want to move calcium against the concentration gradient, it will not go automatically. You have to take help of one molecule which is moving along the concentration gradient. So again the same thing, one of the molecule is moving against the concentration gradient and one of them is moving along the concentration gradient. But again the similar kind of problem. If you want to throw more and more calcium out of the cell, intracellular sodium accumulation will also occur. So what you have to do, you have to throw this sodium out of the cell and throwing of sodium out of the cell is only done by sodium potassium ATPase pump. So again, this sodium potassium ATPase pump will utilize the energy in the form of adenosine triphosphate, and this NCX sodium calcium exchanger is dependent on this sodium potassium ATPase pump. Again, you are finding out the same thing. Although the NCX is an antiporter, but again the antiporter is again dependent on sodium potassium ATPase pump. And what I told you already that if you block this sodium potassium ATPase pump by giving Waven or digitalis type of, right? So what will happen? This NCX will also be inhibited secondarily. Getting my point? So whenever you are blocking the sodium potassium ATPase pump, because the NCX is dependent on that, NCX will also stop working. And if the NCX is not working, what will happen? This calcium will be accumulated more and more inside the myocardium, and that will increase the force of contraction or increase the contractility of the myocardium. And that's why we know digitalis increases the force of myocardial contraction by blocking the sodium potassium ATPase pump. Secondarily, the NCX will also be inhibited in this condition. Right? So this is all about the primary and secondary active transport, and we have given two examples. One is symporter, one is antiporter. Now, in this context, okay, I will clear one more thing, that is, you must have noted that SGLT, generally you have to remember two types of SGLT. One is SGLT1 and one is SGLT2, right? This will be asked in MCQ, very, very famous MCQ, I'm telling you, okay. So SGLT1, where it is present? It is present at the level of GIT, intestinal glucose absorption. It is also present at the level of kidney, but in a specific region of the kidney. There is a part of proximal tubule which is known as proximal straight tubules. Proximal straight tubules. There is one proximal convoluted tubules and there is one which is known as proximal straight tubule. Convoluted first, followed by a straight portion, straight, without any convolution, that is known as proximal straight tubules. Remember, the proximal convoluted tubules contain this SGLT2. So, proximal convoluted tubules contain the SGLT2, which absorb 90% of the glucose from kidney, and only 10% of glucose in the kidney is absorbed by SGLT1. But for intestine, this SGLT1 is absorbing 100% of glucose from lumen to intestinal epithelium cell. Right. Another thing. For SGLT1, okay, for SGLT1, suppose this is SGLT1, okay, so it is absorbing glucose, it is absorbing sodium, okay, absorbing glucose and absorbing sodium. This is from lumen, okay. On the basolateral aspect, this glucose will enter in the blood by a transporter that is known as GLUT, GLUT1, right? So please remember, if it is SGLT1 on luminal side, if it is SGLT1 on luminal membrane, then the basolateral membrane contain corresponding GLUT, that is GLUT1. So 1 SGLT1, GLUT1. SGLT1 on luminal side, GLUT1 is on basolateral side. And if you look at the SGLT2, opposite thing will happen. Yes. So if you think of SGLT2, again it will absorb glucose along with sodium So this is SGLT2. On the luminal aspect, on the basolateral aspect, the transporter for glucose is GLUT, GLUT2, right? So this is true for intestinal epithelium cell also as well as kidney cell also. If it is SGLT1, then corresponding GLUT1 will be there. And if it is SGLT2, then corresponding GLUT2 will be present. But GLUT2 is not present on intestine. Intestine contain only SGLT1. Now there is another thing which is known as the coupling ratio. Coupling ratio. Like for example, if I ask you sodium potassium ATPase pump, we all know that sodium potassium ATPase pump moves 3 molecules of sodium and 2 molecules of potassium. So the coupling ratio of sodium potassium ATPase pump is 3:2. In case of SGLT1, the coupling ratio is 2:1, means 2 molecules of sodium for 1 molecule of glucose. In case of SGLT2, the coupling ratio is 1:1. Oh my god, multiple things, right? So if it is SGLT1, the coupling ratio is 2:1, 2 molecules of sodium, 1 molecule of glucose. If it is SGLT2, then coupling ratio is 1:1, 1 molecule of glucose, 1 molecule of sodium, right? So all these things are very, very important for your MCQ. You have to remember, okay? And some of you may ask that what is the coupling ratio of this NCX transporter, sir? NCX, you told here, okay? Sodium calcium exchanger, okay? For NCX, 3 molecules of sodium will be transported for 1 molecule of calcium, means coupling ratio for NCX is 3:1. Coupling ratio for NCX is 3:1. So this is all about your primary active transporter and secondary active transporter that we have discussed, okay?